Today I'm going to do an update about the course and things to be submitted, because now you have like 3 things to think about at the same time. And then we're going to continue what we did last lecture. So I have like 10 slides left, but they're pretty straightforward, so it should be quite simple. And then we'll get into today's topic. Okay? So clarifications, because I was not sure about the date. So homework 3 is due next Wednesday, October 2nd, before midnight, so 11:59. It's not due Friday, like I said. It's just I had a mistake. The way I did my course outline, I changed it so many times that I got mixed up in the dates, but it's Wednesday, October 2nd. Okay, shouldn't be too much work. Just figure out the gene and then read the paper and quickly summarize what the hell they did. Obviously, if you looked for plant gene or for whatever other organism than human, it's okay. Just keep it like that, as long as you tell me what's the first autosome to be, the first gene to be mapped and not sequenced to the autosome. So be careful with that, okay? Essay number 1 is due on October 11th. That's 2 weeks from now. So if, I don't know, I haven't gotten any question about the paper or any plan or any, is everything going well, or you're delaying it? Because it's a big paper. So, well, it's not big, it's 10 pages, but 10 pages, it's a lot for this level. So you have no questions at all? Or still trying to figure out when to read it? Yeah. Yeah. You want me to explain the conclusion of the paper? Methods? I will explain it, but I'm doing that right after you submit it. So that's the thing, because I want to see how you can understand things there. Obviously, this is like this for any paper you'll read. If you haven't done that technique 10,000 times yourself, you'll have an issue with it. I still, the stuff that I use in the lab, I still read the method and still don't get it, because there's so many modifications you can bring to the same CRISPR protocol, right? And so obviously the methods are going to be super hard to know, especially if you don't do research or you're not in the lab, but don't worry too much about that. As long as you understand the general idea about the method, what I'm interested more into is the scientific thinking of the researchers and the results they found, and if it's supported or not, etc. Okay, so don't worry about the technicalities of what buffer they used and things like this. That's not the point here. Okay, good. Team presentations. 1 person from each team. So everyone has a team? Okay. So 1 person from each team send me an email with the name of the 3 members. Only 1 team did this, so guys, let's go. So I can start already coordinating this. When you have done that, talk to each other, figure out what paper you want to present, and then send it to me so I can make sure that the paper is not, out of scope or something like this, okay? So try to do this somewhat soon. There's no deadline really, but, time is going fast, so get to it. Questions? Okay, so just a quick review of what we did last lecture. So we looked at the process through which you're able to build your genomic library to then sequence this library, which is representative of your double-stranded genomic DNA that you extract from multiple people. So there's 2 things that we talked a lot about last lecture, which is how to produce the genomic fragments in terms of size, composition, and the methods used for that, and the considerations that you had to undertake to get the right DNA fragments. Because if you go from yeast to human, you have to kind of add a level of complexity into the equation, and that changes your way of doing this, okay? And then obviously if you have fragments of different shape and size, then you will need different shape and size in terms of plasmid. And so we looked at all the different methods that you could do, the vectors, not the plasmid, sorry. All the different vectors that exist for this purpose, and going from one to the other, looking at their issues and then their pros and cons until we reach the bacterial artificial chromosome, which is the one the Human Genome Project went with. That and the YACS, the yeast artificial chromosome. So to actually build the library, now that you have a plan, you considered all the stuff that you need to consider, then the protocol is quite simple. So you can see how we didn't get into the technique yet. We're just planning, and now it's the time to do it. So there's a lot of thinking that has to be done every time you're doing an experiment, not just, I'm going to test this and that, especially for a big project like this. So last lecture, we covered point 2 and 4, which is how to fragment the DNA to get a specific size, and how to insert it into a proper vector. So if it's a physical shearing, then you have to repair the ends and add the linkers because it's a blunt end, and that's not good for plasmids or vectors. If it's restriction enzyme shearing, then that's okay. You can just go from there. Today, we're going to look at the last 2, which is how to purify the high-quality human DNA, because you want human DNA that is you don't want any type of human DNA, okay? You want some good human DNA, which is long in terms of size and not have any breakdown or whatever during your process of purification, because that can lead to mistakes. And in a big project like this, if you accumulate mistakes at every step, it just compounds to basically you being so off in terms of your genome sequence, okay? So this is actually very important, and you'll see that there are a few ways to do this. And we'll also take a look at how to fractionate those fragments onto a pulse field gel, which is a different gel than you guys are probably aware of, the agarose simple one-way gel. So this is a bit different. And with all that, you should have the full kind of idea about this. At the end of today's lecture, we'll also start the next topic, which is the actual sequencing, okay? Because we still didn't sequence anything right now. We're just preparing the DNA for sequencing. So this is just a slide from last lecture. So obviously this question, we answered it. How do you make sure that all or most of your clones have inserts? And we talked about the toxic gene that you insert inside of the toxic gene. You dephosphorylate the plasmid so it doesn't religate, things like this. My question to you now is, how many clones do you need to cover the human genome? How do you know this? So do you, yeah, Sarah?
First, we need to cover the entire genome. So we need to know things first, like the size of the genome, size of the insert, and coverage depth, and the probability. And then we can do calculations.
The probability, anyone knows what probability she's talking about? Nope. Covered this in lecture 3 or 4. Yes, Maria? That's the distribution, close. Yeah, so the Poisson distribution is the distribution that represents the probabilities and the number of times your genome is covered. Because obviously, if you're trying to purify high-quality human DNA, and you're going to use that to be sequenced, and you're going to cut it in different fragments, not all your plasmids will have the inserts. And some will have the same insert twice, etc. So you need to have a certain genome coverage. One genome is not sufficient. So you need multiple copies of the genome, right? Usually you need a 10-fold genome coverage. And to do that, you need to calculate using the function that I gave you last time. And then from there, you can figure out how many clones you need, because that will tell you how many fragments you'll have, etc. Okay? So very abstract question, but just to see if you remember a little bit from last lecture. Okay? Good. So this here, I think I said it. Just one important point that I want to cover is the fact that genomic DNA molecules are very long. And because they are long, they can break very easily. So obviously, you don't want to have breakage that happens, because that leads to unpredictable sequencing. You want to keep your molecules the same size. And so because you want them to be all the same size, you don't want your DNA to break pre-cutting. Okay? You want to keep it as healthy as possible. And the previous methods that were used could only yield DNA molecules that are 100 kb. It could work. We could do it with 100 kb fragments, but it will take a lot more time than it should. And so you need a different method to purify the DNA. Which gets me to this point. two techniques exist. So the liquid form extraction. So this is in the past, okay? Now there's a lot of more techniques that exist. But in the past, you had the liquid form extraction. So basically, you have your tube with just a liquid in it, and your DNA is kind of soluble in it. And that's where you put your cells, put some detergent, break the cell, the nucleus, etc., until you get to the DNA, okay? So this is the liquid form extraction. Usually you use phenol chloroform, so it's highly toxic, but produces, especially chloroform, and it produces DNA in the range of 50 to 200 kb. What's preferable for our project, the Human Genome Project, is to use these what's called agarose plugs, and these are just blocks of agarose. And they're interesting because when you put the DNA in the blocks, in these plugs, what happens is that they stick to it, and the DNA is kind of protected from shearing. And so what you do is you take your single cells, one cell, you put them in the agarose plug, and then you remove the proteins and lipids, and you can do this through a series of washes, right? Different macromolecules are soluble in different solvents, and so if you use the right solvents, then you'll remove proteins in one go, and then lipids in the other go, and then you keep going until you have only DNA. What's cool about these plugs is the fact that you can do everything in that block, and including the enzymatic digests, and these produce DNA of about 1 million bases. So this is pretty good for us. Now, obviously, I'm saying a lot of numbers here, right? Just remember that the DNA needs to be about, the bigger the better, so higher than 350 kb is what we're interested in, okay? This is what the bag does. But if you have 1,000, if you decide to go with 1 million base pairs, you could still do that, okay? The bags can take up more DNA. It's just their optimal point is 350 kb. Right. So here we're talking about the Human Genome Project, but in reality, depending on the project that you're working in, there are a few rules that you will want to follow. So if you're choosing which method to extract DNA with, then if your genome is quite small, then you'll just want to go through the liquid form extraction, right? But if it's pretty big, then you'll use these agarose plugs. And usually, when you're thinking about the size of your fragments, the size of the DNA molecule that you're trying to fragment needs to be about 5 times larger than your clonable fragment, okay? So if you have 1 million base pairs, then your fragments, you divide it by 5, okay? Very simple like that. And then obviously, because of that, depending on your DNA fragment and the DNA molecule that you're trying to fragment and the size of the fragments, then you'll choose your specific vector system. You will not use a bacterial artificial chromosome for a 5 kb molecule, right? Fragment. Usually you use those for much bigger DNA sequences. Obviously, for BACs and YACs, the agarose embedding method is preferred, and this just makes sense because of the size. Because the bigger the DNA molecule gets, the more chances it has to break, and if you're mixing in the tube, you will probably break it, okay? Good, so that was purifying DNA. Now, size fractionation. So when you break your DNA, and you're optimizing specific conditions at specific time and concentration of the enzyme, you will not always get a uniform distribution of fragments. Obviously, some of them will be bigger, some of them will be smaller. And so because of that, what you need to do is use a gel to separate them based on their size. So you can imagine if you have a piece like this, and then you break it into 2, right? Sometimes, so let's say you break it right in the middle, you have 2 pieces, right? So what happens is that maybe one piece is too small, and you will have to ignore it because it's not 100 kb and higher, for example. So this is why you need a higher genome coverage, because in the other, if you cover the genome more times, maybe in the second genome coverage, the break is at a different part, and will give you a fragment that includes that 100 kb piece. Does that make sense? So this is why you need to cover the genome multiple times and not just once, because breaks don't happen always at the same place, and sometimes fragments end up being over-digested. And because they're over-digested, they don't fit in your criteria of the size of the fragment. And so they're removed from the equation. So if you have multiple genome coverages, you'll have more chance to have those fragments that were over-digested, less digested in a second coverage or third coverage or fourth coverage. Make sense? Easy? Okay. Now the gels that you've seen in your labs, in the teaching labs, are not useful for big size like this. For this, you need something called a pulse field gel electrophoresis. And this is a technique where you still use these gels. The only difference is that the current that you're going to apply to it is not in a single way, a single direction, but instead it is in 3 different directions. Why 3 different directions? This will allow the DNA molecule to reorient itself, as you can see here, and it's better for the movement of the molecule. Okay? If the molecule is super huge and you have many of them to separate on a gel, what's going to happen is that if they go at the same speed, it's not linear. It's more like exponential. The smaller you are, the faster you go, and as you become bigger, the force required for 100 and 200, so the same force will displace 2 big pieces at about the same speed, even though they have a difference in molecular weight, okay? And this is just because of the physical properties, which I don't get 100%. But what you can do to separate them better, these big molecules on your gel, is to actually alternate the current using these 4 plates here so that your molecule zigzags like this. And this will add to the length of traveling of each piece, as you can see here, instead of just going straight. Okay? Does that make sense? What else? So alternate current is used here with these different plates, and this allows for better separation of these big pieces. Good. So this is a summary of everything I said prior to this. So these are all the cloning systems that you can use. Standard plasmids, phage mids, cosmids, etc., up to the BAC and YAC. And then this here is not true. They can go up to 4.6 million base pairs because the F plus plasmid of the bacterium, so in the HFR strain, is about 4.6 million, and it can technically transfer it completely. Almost never happens, but this is why they put up to 300 kb. This is like the optimal that you can get, okay? And the copy number per cell, if you remember, we want a lower copy number because we want to avoid rearrangements in the DNA, the human DNA, because that will just mess up the genome more and mess up all our contigs and then get us lost in sequencing, okay? So the less copies, the better. So obviously, as you go down here, you're getting towards what you want. And then obviously, you can also use phages, which I showed. I think it's the first technique that I showed. Okay, genome coverage considerations. So this was the answer to a previous question. For the human genome, how much do you need to cover how many clones do you need to cover a certain probability of the human genome? So usually if you're doing a sequencing project, you want 10x. De novo sequencing, you want the highest probability. If you're doing for any other purpose, usually 5 times coverage is good. But you want to be highly redundant when you're doing your screens, because when you're highly redundant, you allow for more fragments to be present in your sample. Good. So now the lab stuff. If you want to cover the human genome 10 times for your sequencing of the whole genome, you will need about, if your fragments are 50 kb, you'll need 600,000 clones. If your fragments are 3 kb, you need 10 million clones. So this seems like they're just little bacteria, so it's okay, but actually this is a lot, okay? It's, you need about 26,384 well plates, and I'm pretty sure that we can't fit this amount in all the freezers that we have in the biology department, okay? So this is crazy. This is why you need bigger fragments, because then you decrease the number of clones and you decrease the amount of work that needs to be done by researchers. Okay, so you can see how big this project is. Not sure how many it is for, let's say, 250, but you can calculate that because you learned that. Good. Any questions up to now? Simple? Okay. So we built our library of, I don't know how many million clones. Now, actually, it's less than, it's probably like 100 or 50,000 clones, something like this, okay? So now we need to map it, and we need to assemble it in the specific order that it is in the DNA, so that then when we sequence it, we can get the full sequence. And obviously, these segments are not in order. Your clones, you don't know which one comes first, which one comes next. So you have to figure out some sort of way to map them. And this is what we'll be talking about today. This is, again, just to highlight this, it's for the Human Genome Project. Celera's method did not do this. They went directly from this to this. They skipped number 3. Okay. We'll see how they did that. So the goal is to take your DNA, part of your chromosome, with your GIM sustaining or whatever that you want to analyze, and then you will cut it into fragments, and then your goal is to get all these fragments in a specific order that then you can sequence, and when you sequence it, you will remove these overlapping parts to kind of get the full sequence, okay? And this is done by computers. So there's a few things that you can do. These are your contigs, basically. So there are a few things that you can do. There's 2 methods that are used for this purpose. Obviously, scientists are very funny with the names, but chromosome walking and jumping. This one here is quite not efficient because it takes a lot of time. So this method is very old. It's one of the first strategies that was thought of, and it's slow because it is sequential. So that means that you need to sequence a segment to then have the ability to sequence the next one, and the next one, and the next one. You can't do it in parallel, let's say. And so if you want to sequence, let's say, for example, this blue one and this purple one, you have to sequence all this part here, okay? And so you need to, and so the way it works is that you have to first start with a starter clone, take any bacteria that you have an insert in, add linkers to it, a primer that binds those linkers, and then use that to sequence that first piece. When you get the full piece, then you will put a primer at the other side, sequence that part, and then use that as a primer for your next segment, right? Then when you finish that, you put another primer at the end of the sequence that you just got, and then you find another clone, and then you keep going, right? So you need to basically put a first primer that is linked to a linker, get the sequence of that first fragment. When you get the sequence of that fragment, produce a primer that is that hybridizes with the end of that fragment that you now have. Because you have its sequence, then you can get the primer. Use that primer, build a fluorophore, tag it with a fluorophore, put it into your solutions, find the other clone or all the other clones that have that sequence, and then do the same thing. Add an adapter. Actually, you don't need an adapter now. Use that primer to sequence that fragment. Make a primer using that sequence onto the next. Make sense? Kind of? Questions? No questions. Wow. Yes.
It says it goes in both directions, so why is it that when you have to sequence the blue and the purple one, you need to sequence the middle ones?
The blue and the purple?
You said that you need to sequence all of them to get the blue and the purple one?
Yeah.
But if it goes in both directions, can't you just like start and stop?
Because imagine your chromosome like this, and you cut it into multiple pieces. So if you go from your first piece this way, there's nothing. And for you to get to the other end, you need to go through everything because you don't know where you are, right? And so in your fragments here, I'm showing a very simple example where you're at the edge. But your first clone that you could pick could be right in the middle of your chromosome, right? So that first fragment, you will add a linker to it on this side, sequence that part, and then at the end, you'll have the sequence, make a primer, and then go, primer, go, until you get to the end. There's no way for you to go from here to here using this method. Do you see some way that you could do it? No? Does it make sense what I'm saying? Good. Just should be quite clear. If it's not, you can ask me about it. When you have these clones, obviously, when you have your fragments are not all the same size, right? But they're not of the same composition. So because they are not of the same composition, you will have different sequences, even though it's the same size. And so for you to now map them, you need to break them using more restriction enzymes, different restriction enzymes, to get fragments of different sizes. I'll show you this at the end. This is just for step 2, okay? But for now, the one that you need to understand at this point of the lecture is this one here. So each clone gives you the next clone, and then from there you go until you get all the subclones starting from this point, okay? Now, how do you identify your clones? If you remember in lecture 4, I think, I showed you this technique called the colony hybridization. Now, in this diagram here, you have a plate with all your bacteria that have the plasmids that have your inserts. And so you would put a nitrocellulose membrane on top of the bacteria that will transfer a little bit of bacteria from the master plate. And then what you do is you come to this step and you break the cells, you wash, you use different detergents, things like this, and you fix the DNA so it does not move from the membrane. Then you use your probe, which is a primer that is attached to either a fluorophore or that is radioactive or whatever, and you will scan it under a specific machine that tells you which colonies react to that probe. Because those colonies react to that probe, that means they have an insert in their plasmids that has that sequence. And so if, for example, you're taking a look at this one here, because you sequenced the first part, you got this primer here by taking the sequence and then building your fluorophore from it. So it's just a primer with, let's say, 32-phosphate, radioactive phosphate. Then you go back to your master plate, your nitrocellulose membrane, and then you add, let's say in a bag or whatever, your fluorophore conjugated primer, and it will give you 2 colonies, for example. One that has this insert and one that has this insert. Does that make sense? So then you can match all the colonies and which ones have similar sequences between them, and that will tell you which ones are your contiguous sequences, okay? Now remember one thing, they're all the same size. So your fragments will be the same under the gel, right? All you know at this step is that these 2 fragments or these 2 colonies that you just did the colony hybridization have the same sequence, but they could be two different parts, or they could be the same exact fragment because your genome coverage is 10 times, let's say. Make sense? So we'll see in a bit how we can actually differentiate the same size fragments into, which one is which, okay? So this method here is just for plates, but in our case, if you remember, we need a lot of 384 well plate, because if you do it with a plate like this, it will never be done, okay? So you use these 384 plates, which look like this, and each of these holes here has one of the colonies, okay? And one of the clones. So you don't know what it is, and so you can imagine stacks of this, and then you have to figure out which ones go with which. So every time you have to stain, extract DNA, stain, match the clones, sequence those. No, not even sequence, just put them in contiguous assembly, and then repeat until you do the 3 billion base pairs. This is crazy, okay? This is a lot of work. Just this one plate, I think I've done maybe like 10 in my whole life, okay? Because it's so much. Like you just pipetting the whole day, and it takes hours to just do one of them. Now imagine you have to extract DNA, hybridize, match clones, repeat, and then you keep doing that for 3,000 plates. Crazy, okay? The way you transfer your bacteria is using this black and white picture because it's super old. This pin, what is it called already? A pin replicator, which has 384 pins. And then what you do is that you go on your lawn of bacteria and then transfer it in the plate. And you try to get unique bacteria, unique colonies each time. Sometimes you'll be having duplicate colonies, which are the same thing, because your lawn of bacteria is not super, your bacteria are not spread evenly, and so you will have duplicate clones, which basically are not telling you anything extra. Okay, so a lot of junk data, let's say. Does that make sense? Good. So it looks like this. For the human genome, 17,664 human yak clones, so that's yeast artificial chromosome with inserts, are blotted on this membrane only, okay? And then each square contains 36 clone units, so this is just to tell you how big this project is, and this is just for this membrane. There's a lot more, okay? And so what you do is that you blot your clones twice, and then you do that because you want to avoid false positive data. And so let's say, for example, you have your primer with a radio-labeled 32-phosphate. Then you will put it into a bag with your blot, your membrane, your nitrocellulose membrane or whatever membrane you're using. And then it will tell you which clones are kind of contiguous, or they have that sequence, right? So you can see here that these ones showed up, this one popped up, and this one too, these two here, this one here, and so it's telling you that these have the same sequence that reacts to your probe, which is your primer with 32-phosphate, okay? Now you will take all these, and this is one of your contiguous parts, okay? Some of them are the same, the exact same fragment, others are not. So you have to figure that out, and we'll see how we do that. Now, why do you do it in duplicates? Because your probe, sometimes DNA is sticky, so sometimes your probe can interact with your fragment or a different fragment just because life, okay? And so the chance of it binding twice by mistake is rare, and so you do it duplicate or triplicate to avoid false positive data. So if you can see here, there's only one dot here, so that means probably this is a false positive. So this is how you decrease kind of the rate of false positives. Okay? Take a break. Talking super fast here. One way to improve this, so here we were acting kind of blindly. So you're adding the linker, sequencing, find the sequence, make a primer from that sequence, and then continue, right? But you can also take advantage of the map that was created like 3 years before that, which is the sequence tagged sites. So these sequence tagged sites, if you remember in lecture 5, I think at the end, there was a map that was made with about 40,000 sequence tagged sites, and these are unique sequences in the genome. Because we had these sequences already from that project, then you can actually use those as your starting point. And that allows you to kind of have a basic map that you can then use to build on by placing all the fragments. Because if you know that this here is an STS, then you can use its sequence to produce a primer, and then use that primer to find the fragment that goes this way and the other fragment that goes this way. And then you can see how you can make a primer for each of the STS and then start hybridizing to the right colonies that have that fragment. Make sense? Questions? I'm going super fast. If you don't ask me questions, I don't know. You good? Okay. Yeah.
Is it possible to make a primer where, because there's so many repeats in the genome, you actually sequence it 10 different sequences that have the same primer?
Yeah. So these sequence tagged sites are unique, but if you're making primers, let's say you're making your primer here, this could be an inverted, this could be a microsatellite, And so you can make a primer and then end up having so many fragments that appear, so many colonies that hybridize with your primer. And so then you end up just turning in circle, because it will bring you to probably a different chromosome, and then you start looking at that specific fragment, and that fragment brings you to another chromosome, and so you get lost. Okay, so you have to avoid the repeats, and this is why we're going to talk about the second method, which is chromosome jumping, but we're not there yet. Okay, but very good question. It's an interesting point. This is pretty genius. So how do you avoid staining all the plates that you have every time to find which clones work together? So what you do, let's say you have all these plates, then this is called your master pool. So all your plates together, this is, let's say, 864 clones. Forget about this here, but basically it's telling you where which clones react to your probe. But what we're interested in is more this part here. So one way to do this that is very simple is that you will take all your plates and you'll pool each row in a tube by itself, and you'll pull each column in a tube by itself, and then you'll pull each plate in a tube, right? So then you end up with, let's say, if you have, I don't know, let's say 10, if you have 10 plates or 9 plates, you can imagine that you only have 9 tubes for your pooling of each plate alone, right? So you will take every sample here, put them all together, so that's pooling one plate, and you'll do the same for each plate, but inside the plate you'll take the rows, so each row has its own tube, so you take all the holes in one row, put them all together, and then the columns you do the same, okay? Make sense? Then what you do, instead of staining every freaking plate, you stain the pools. So first you have your plate pool, which are here. So they contain the fragments from all the plates. So A, for example, this lane A here means that all the clones inside plate A are in this tube. Do the same for B, C, D, up to I, and then you have your row gel. So the column here, A, means that it's all the samples in here together. And then you'll do the same thing for your columns. So let's say number 1 here means it's all the samples in column 1. Now you have only 3 gels to stain, or 3 membranes to stain. When you stain these 3 membranes, it will tell you the plate, the row, and the column that are all hybridizing to the same probe. So in this case, for example, the plus and minus are just your positive and negative control to make sure that everything works. Your gel, your membrane works, your staining works, all that stuff. In this case here, you can see that for this specific probe, plate G at row E, and wait, this thing here is annoying. And column 5 has the clone that hybridizes to that. Does that make sense? Maybe? No? Good? Okay. This way you can just take the same samples and put your probes every time instead of doing, because if you were to do it for all the plates, you would have to put your probe inside each one of the holes for all the plates. And that's just for one probe. So imagine you have thousands of probes. Crazy. So this is much easier, and then you could just repeat the same protocol no matter what probe you're using. Okay? So this is how they were able to save a little bit of time. Problems, because obviously there's a lot of problems. This method here is sequential, so you have to sequence in sequence. You can't, you have to wait for one fragment to be sequenced to get the next one, because you need information from that fragment to get the next fragment. If you get to these repetitive sequence blocks, so blocks of DNA that have the same sequence, then you end up having an issue where you think you're walking, but actually you're going to another chromosome or whatever. So these repetitive sequences that are found a lot in the human genome have to be avoided. and we'll see how we can avoid that. Obviously, yaks and backs, you can have deletions and rearrangements and all that, so your map is not as good as it can be. And this is very important that people don't realize this, but genes that are in the human genes can be toxic for bacteria, okay? So depending on the protein that is being made, it can actually trigger things in the bacterium and then, or not even go inside the bacterium because it's too big. So obviously bacteria and human cells have different translation and transcription. For example, no introns in bacteria, but still, just that sequence can be toxic to the bacterium. And you can take a look at MYC, MYC, the MYC gene, if you're interested in understanding this. But now they're using bacteria to basically take care of MYC cancers, so MYC-dependent cancers. So this is some crazy stuff. You infect actually the person to cure her. And so this is just because of how proteins and genes in mammalian cells react in bacteria, right? And this is the same for yeast, etc. They trigger different things. And what's important to understand is that even though maybe they don't have the pathway, just the sequence can actually bind to something else, right? So this is the issue. There's not many examples of this, but obviously there are some things that are toxic for the bacterium. Chromosome jumping. So this is the second method that basically aims at just avoiding those repeats. It was made, was kind of used by Francis Collins. So this is the director of the Human Genome Project, also the scientific advisor of the current US president, also the guy that cloned the cystic fibrosis gene, the muscular dystrophy gene, and so many other things. So this guy is a G. He used this method to clone this gene, so the CFTR gene, which creates this ion pump, if I'm not wrong, in your lungs, and forgot where else, and that just creates problems with your mucus production. Not important for this course, but I think this disease is quite interesting. I don't know if anyone knows what cystic fibrosis is. You heard it, or you actually know what it is. If you've ever seen those, usually it's old people, but sometimes babies too, they have these that thing that you breathe with. In French, it's easier, respirateur, right? And you put them here, and they're walking with their like oxygen tank there in the bus or whatever. That's basically that issue. It's because the mucus prevents them from taking up oxygen, okay? And this is quite dangerous. So this guy was able to clone this gene. Why did he use this method? It's because the CFTR gene is actually about 250 kb. So it's super big. It's one of the biggest genes that you have in the human genome. And because of that, you need different methods of cloning it. I'm not sure if you got the Nobel Prize, probably not. This disease is quite interesting because there's one way to fix it, gene editing. So you can just remove the broken gene. Actually, you don't even need to touch the whole gene. Usually it's like one base pair or two base pairs that are messed up. But obviously there's clinical research that's being done for that. And as you've seen an example of this at the beginning of the course, so stem cell therapy with modified cells was able to cure that patient of diabetes type 1. Now you could do something similar with this and modify the cells of that person to actually have the right copy of the gene, and then that will save them from cystic fibrosis. That's not the purpose of the course though. This is just the technique that was used, so I'll tell you a bit about it. It's similar to a method that's called paired end reads, and this is a method that is still used right now. But basically chromosome jumping is the grandparent of this technique, let's say. It's the very old way of doing it, and it lets you sequence sequences that are further apart than 40 kb, so you can jump over the blocks of repetitive sequence. Okay, I need you to focus so I don't lose you. So chromosome jumping, okay? There's a theory let's say in physics, that says if you want to go from this point to this point, the fastest way is not like this, but it's to flip the dimensions so that this point reaches here, okay? So this is the same with DNA, because we always think of it as linear, but what if you circularize the DNA? Then those two extremities are now next to each other, right? And you're skipping the part in between, okay? So if you're taking a look at genomic DNA, and you have, let's say, a repetitive block that you are trying to avoid, then you can use this method here. And the way it works is that you'll take a restriction enzyme that does not cut a lot. So they have different cut sizes and efficiencies and all that. SAW3A, for example, is highly efficient. It cuts 250 base pair long on average, not one. I think it's like 500 kb or something like this. So you completely digest the genome with this, and what's going to happen is that you'll have this little fragment like this. You can then ligate it together using a ligase, and it will circularize your molecule, right? And so in this case, you can sequence two parts that are separated by a block of repetitive sequence. So if you spot that repetitive sequence, then you go back in your samples and then cut the molecule and then just ligate it back together so that you bring these two pieces closer to each other and you skip all the repetitive block here, okay? And you can do a lot of this. You can sequence, yeah?
How can we know if it's a repetitive sequence if we haven't sequenced it yet?
Right. That's a very good point. I'm presenting this technique now, but obviously this is talking about sequencing. So this is used at the sequencing step, okay? So you're building your contigs using this here, right? And then you're adding to it this method here. This method here, what it tells you, so for example, let's say you have a clone, you made a primer, and your primer is a repetitive sequence itself, right? And then you get to another fragment, and it's the same repetitive sequence, right? And then you hybridize it, and you see the same colonies being, right? So you need to kind of change your way of doing it, right? So you do this, but obviously this is really interesting in terms of sequencing, right? But to build your contigs, you can also use this. Make sense? Good question. Okay, let me just write the names here because now there's a lot of people. One second. So Alessandra, Maria, Sarah, Arthur. Who else? Gabrielle. Anyone else? Shauna. And I think that's it. Okay. Yeah, that's it. Okay, next step. Let's say you have 20 or 2,000 fragments that are 100 kb long each. Now, how do you start mapping them? That's the question, right? Because even though you're building your contigs, you just know which fragment goes with which, but you're not sure in which direction they are, right? And so what you do is that you need to find your overlapping clones. So now each fragment that has 100,000 kb, for example, you will use restriction enzymes to cut it even more. And then you will do this technique called restriction fingerprinting, which is basically breaking each fragment, each insert that you had in your plasmid. You have to break it in multiple pieces using 1 or 2 or 3 restriction enzymes. And that will give you a specific banding pattern for that fragment. Now, let's say you have 2 fragments here that you're trying to map, see if they are overlapping or not, etc. Because now we're talking about jumping, right? So if you're jumping far from there, how do you put things into perspective? So you need to break everything even more. And each one will have a specific fragmentation pattern. Because even though the fragment is 100 kb long, the sequence itself is not the same between 200 kb long fragments. And so the restrictions enzyme cut sites are not the same. And because they are not the same, they will give you different banding pattern on a gel. Usually you use a combination of 2 or 3 restriction enzymes to get a higher number of fragments so that you can create a unique pattern for a specific one. Okay? Obviously, if you see that there is a band or two bands that are the same on two clones, on two fragments, then you know that those two are overlapping because you use the same set of restriction enzymes and they gave you the same bands. So that means that fragment 3 and 4, for example here, are shared by the two fragments. Okay? Make sense? Yeah.
And why do we do that? After two sequences, just one time the overlapping sequence of names?
Sorry?
Why do we want to identify overlapping clones?
To build your sequence of contigs, right? This thing here, because you're jumping, right? You're walking sometimes, and then you're jumping also. So if you're walking, obviously it seems obvious. You're just gonna go like this, and it makes sense. But you're also jumping, and so because you're jumping, then you create a higher level of complexity, and so you need to find a way to map those jumps, right? So it goes by this. So you will break all the fragments separately and then build backwards from that. Yeah?
This creates a bias.
Sorry?
This creates a bias for like these overlapping regions.
A bias in terms of what?
If your reads are overlapping, so you would have like another good read, then you use this to define these regions.
Wait, I don't get it. Say.
Like you have these reads.
Yeah.
They're overlapping.
Yeah.
You create a bias, and you use this to like make those errors right to find.
Exactly. So your bias is the fact that, well, you have many biases in this case. First, your fragments are all the same size. So you don't know how they're placed together. So if you're walking, you can find them all, but then sometimes you have to jump, right? So that's another issue. And because you have to jump, you have to figure out what goes with what, right? And so you have to go backwards by doing this. Does that kind of answer or no? It is not a good way of doing it. Do you have a better way? Well, there's Craig's Venter's way, which is just break everything and then sequence each piece separately, and that's it. Makes sense? Is it a better way? Oh, you're doing it more than twice. We're going to break them again. You're breaking them again. Yeah, it is. It is highly inefficient. The thing is, one thing that's interesting about this is the fact that you're using DNA from a lot of people. So if there's deletions between two different groups, you need to be able to account for that. This is why this is called hierarchical. So it tells you if there's a deletion or not. Craig Venter, even though he's like a really good scientist, he took his own DNA and sequenced it. But his own DNA does not mean that we have like the same deletions, inversions, translocations. And so that method is not perfect. This has biases. This has a lot of work more to be done, but it gives you a more generalized sequence of the human genome because it accounts for the inversions, translocations, things like this, okay? But we'll cut them even more. How can you do restriction fingerprinting? So we're talking about, so these are some considerations. We're talking about DNA size now. Because we're talking about DNA size and cutting even further, then it needs to be highly precise. And that for a gel is quite complex. And so, because you can't just say, oh, this fragment is between 100 and 120 kb. It's like which one it is, because those sizes are important for placing all these for the fragment, okay? So if you cut it, if you're 100 kb, you cut it and you get 4 fragments. And let's say, for example, one of them is 25 kb. Then you need to know that it is 25 kb, because then you'll use that to find other 25 kb fragments using those enzymes, and then you know that those sequences are potentially overlapping. Now, obviously, you can take a look at the gel like this and say, oh, this one is the same height as the other one, and so that means they're in the same clone, but this is 2 lanes, not thousands of lanes, okay? So you work with specific sizes instead of taking the whole thing and then using a ruler and like, okay, this is the right one. Does that make sense? Yeah.
If we have a repetitive sequence, then the restriction enzymes are going to cut like regularly, and then this we're going to see that it's the same size, so we can say it's overlapping sequence, but it's not.
Exactly.
So how do we do in this case?
How do we what? Sorry?
How do we do in this case when we think it's overlapping, but it's not in reality?
When we think it's overlapping, but it's not.
Like in reality, it's repetitive sequence, but with this method, we can think it's.
Yeah, I understand. Let me think. Very good question. Because they are repetitive sequences, then you'll end up having same-sized fragments. Let's say if you cut, and then if you use different sets, that's a very good question. I'm not sure. I have to think about it because, yeah, I'm not sure. I have to check, but I'll get back to you next class. I just have to sit down and draw it and go over it or ask ChatGPT, you know. Okay, so to increase the accuracy and to be able to predict the size precisely, you will use DNA-sized markers that you will put on the first lane of the gel. But you don't do it only for the first lane. You load it at every 5 wells. And you don't use only one DNA-sized marker. You use a combination of 3. So this way you have a band density for the marker at every, let's say, 5 kb or something like this. Okay? This way it allows you to predict more precisely. So first thing, you load your samples in triplicates, obviously, to make sure that there aren't any problems during the process. And you automate the analysis because doing this by yourself is a lot. Because the gel looks like this for, let's say, 1, 2, 3, there's maybe like 10 fragments here. Okay, you see the triplicates here, and then the triplicates here, etc. Okay, so this is your ladder. This is another ladder, and they have 3 different markers in it, not a single one. And then you have to make sense of all this to find your contiguous sequences. Those that have the same, like for example here, right? So these two might be overlapping, or they might just have the same sequence, but they're not really overlapping, okay? So it's the specific pattern that tells you that, and I'll tell you how you do it, okay? So each lane you have to image it, you have to take a picture of it, so this is the automation part. And then you will use that, and you'll compare the image to the ladder that you have on that specific gel at that specific time. And just the distance will tell you, like let's say, for example, this fragment here is 1 centimeter away from the top. And let's say, for example, this one is also 1 centimeter from the top. Then you know that it's 12,000, kb or whatever. Okay? And so you will do that, and you will get all the sizes for the bands that you have cut using these different enzymes, okay? So it gives you all these sizes, these sub-fragments of the main fragments, okay? And then you have to compare it against the restriction fingerprints of other clones. So you do the same thing for all clones, and then you compare them. And you'll see some fragments are the same, others are not. So this potentially could tell you that they do overlap, right? And then they're saying you do a database search because you can imagine that you have, let's say, for example, 10,000 clones, and each one of them have their own fragmentation pattern. So you need to do some sort of database search to align the different fragments between each of the clones together. You do this using computers because otherwise it would be too much work. Okay, we could make sense of this, but it's easier to use a computer. So then let's say, for example, you search for more clones that have fragments in relationship to this one, so similar to this one. Then you might find clone number C, which clone number C has these fragments, but not others which are shared between A and B. So this is based only on the size. And then you can build your contigs like this. How do the repetitive sequences come into play in here? I have to think about it, okay? Does this make sense? Kind of. Not sure. Yes?
So if we would have found a match between A, B, and C, and
Yeah.
theoretically, that means that A and C are also overlapping.
Exact. If you find match between A, B, and C, that means one of them overlaps 2, and the other 2, each one overlaps that one. So let's explain it. Let's explain it in using only 2. Should I use the board? Maybe. I don't know if there is a chalk, but you know what? Let's do that. Switch it up a little bit here. Okay. Let's do this. Okay, let's make it like super simple so you can get it. So imagine just 2 fragments, okay? We're going here. And you have your 2 main fragments that you inserted in your bacterium, okay? And you used enzyme, I don't know, X to cut it, okay? So this gives you 2 fragments that each you have to break again, right? And if you break it with, yeah, so let's say, for example, you take this one here, then you need to figure out how the hell is it, actually I will do 3 so it's easier to get. So if you have 3 fragments like this, your goal is to understand how are they linked to each other, and that will not be cut exactly equal like this. You'll have actually some that will be cut like this, and then some that will be cut like this, and then like this, right? Your enzymes can cut here, but sometimes they can cut here, right? So you have multiple spots. Does that make sense up to now? Okay, depending on how you digested your fragments. So now to figure out how they are ordered into a contiguous sequence, what you need to do is break each of these pieces into smaller pieces, right? And so you'll cut them at different spots, and then you'll cut this one, for example, I don't know, here and here. And then this is using a different set of enzymes. So let's say Y, Z, and then, I don't know, Y, Z, Y, Z, or something like this, okay? Just the same set of the same set, let me just do it like this, the same set of enzymes. And so what you realize is that when you cut this fragment here, you'll have a piece that is, let's say, just random examples, okay? 1 kb. And this piece here that, you have to do it properly so that you get it. Sorry, guys. That is exactly 1 kb here, okay? Using the same set of enzymes. So you know that these 2 pieces are of the same size, okay? Because they are of the same size, and let's say, for example, all your other pieces are different sizes. Let's say this one is 3, 5, 7, 8, 12, etc. Your only 1 kb piece is this and this, right? Because on your gel, let's say you're putting all your, you're putting all your 3 fragments on the gel, so that's fragment A, B, and C. And at 1 kb, there's only A and B that have that 1 kb band. That means you know that these 2 are overlapping. Because with that specific set of enzymes, you got only these 2. That means if you get, for example, I don't know, another band here, so that means now the 3 of them are overlapping, and then obviously there's one of them that is at the center of both, okay? So it would not be a pattern like this. It would be more a pattern, let's say, like this, where you have this one overlapping with the other 2, okay? Now, obviously, this is a simple example. If you do it for the human genome, then you end up with all that. Does that make sense? Cool. I have to, yeah?
So it's just A, for example, it's just A and C overlapping B, but not A and C.
Sorry, can you repeat that? The first example?
So like there are 3 fragments, so like only the middle one is overlapping the other 2, not the...
Exact. But in this, yeah, go ahead.
Why do we say that all 3 of them are overlapping? Why not just A and B and B and C?
Wait, so I gave 2 examples here. The first example I gave is where only A and B are overlapping. So that was the example where this was not there, and you had this one, and then this one, right? So let's assume this is fragment A, this is B, and this is C, okay? So here you can see that A and B are overlapping because it's the only two that when you break them with the enzymes, you get a 1 kb fragment. Now, obviously, if you compare these two, maybe this part here is, let's say, 2 kb, right? And then at 2 kb, what you see is fragment B and fragment C that have these subfragments at 2 kb. So then you can say B and C are overlapping. But if you see, for example, a fragment that is shared by all three, how is that possible? Okay, so this is 1 kb, this is 1 kb, this is 1 kb, and this is 1 kb, right? So then we're not at the sequence yet. We're not at the sequence. Yeah, we're just looking at how we map it in relationship to each other. And obviously, this is a very simple example that's using only this. But with the whole combination of different fragments that you produce, there's a pattern that is unique, right? Obviously, there are mistakes that can happen here because it's not precise. As you can see, they're not the same sequence, but they're still overlapping, right? So when we get to the sequencing, then, okay? Does that make sense? Kind of. A little bit complicated today. I'll do a review next lecture, give you the time to process it, and then I'll explain it. But we're not done. Where are you guys going? So there's a problem, as we are seeing here. So you can have duplicated sequences, and because you have duplicated sequences, they fragment the same way. Because they fragment the same way, then they can mislead you into thinking that two fragments are overlapping. But they're not. So that's the first step. Obviously, this I mentioned earlier, but today it seemed like less, let's say, fluent because there's so many issues with this approach. The human genome is not use one method and get the whole genome, right? Because it's so complex and there's so much variability. You have an approach, but there are special situations where you need special solutions. So it was not just done like what I'm explaining here, where you just walk the chromosome and sometimes you jump it and put everything into similar sizes. Actually, this here is quite complicated. And so sometimes you can end up thinking that you're going from one fragment to the other, but actually because the sequence resembles the sequence from another place, then you are diverging, and because you're diverging, you'll start kind of assembling between different chromosomes, right? But you're not going the right way. It's not the same sequence. It's not the same chromosome. You're like going somewhere else, right? So obviously for these different situations, you need more refined work, and you can't just use this and say that this is it. There are a lot of things that were done to solve these problems, and they're mentioned in the paper. But in general, this is how you would do it. So just a quick review, and then we can go. You have your chromosome broken into same size fragments, put them in your bacteria, and then you have your library of DNA. Then you use radioactively labeled probes to find sequences that react to the same probes. That means those sequences are either the same sequence in two different clones because your genome coverage is 10x, or it's a sequence that overlaps with another sequence. And then for you to figure out how they overlap or where they overlap, you will have to break it into smaller pieces using a different set of restriction enzymes, which gives you a unique pattern of bands on the DNA for each fragment because they have a unique sequence. Each one of them, they're not the same. And because of that, you can see that, for example, in that example, if fragment A and B have a 1 kb fragment, but no one else has that 1 kb fragment, That means that that 1 KB fragment is shared by both. Cool. What else? What else? Obviously, if you have duplicated sequence or your microsatellites or all that, then obviously that does not apply anymore, because duplicate sequence can mislead you and bring you to assemble a contig that is not of the same chromosome, and so you could be jumping to another chromosome. Okay? And this is where you need special solutions for this, like separating the chromosomes, etc. Okay? Using the sequence tagged sites, which are unique sequences, to overlay that map with your contig assembly. Okay? No time. But next lecture, we'll take a look at how we sequence the actual fragments, and you'll see that we'll break them even more. And so, right before you guys go, I think you have the knowledge to answer this. If we're sequencing at an even smaller level, because the sequencing reaction is not as efficient, you can't take a 100 KB fragment and then sequence it in one go. Usually it goes for 600 bases and then it stops. So why not go directly to 600 bases fragments instead of developing these contigs and using the backs and all that? Sarah, you asked the question, so now it's time to answer it, no? Yes, June.
I'm not sure, but it is because since this was just a little preview into a tiny subset of an entire genome, and if we want to work on entire genomes, doing it like this is going to be horribly inefficient. So we need to take a larger scale approach, which is why we need contigs and fragments.
Exactly. So 99% true. There's another team that did something different. How were they able to achieve that? No clue? Okay, that's the topic of next lecture then. So I'll just explain it to you quickly. If you're trying to make a map of a 1 million base pair chromosome, you're not going to cut it in 600 and then find overlapping pieces between those 600 bases. What you do is that you cut it in huge pieces, let's say 10 pieces of 100 KB, which overlap. Then you know how they are arranged on the chromosome. Then you can fine-tune inside those segments. But if you go directly to the small fragments, the complexity that adds is that now you have billions of or millions of 600 basis fragments, and you need to make a contig from those. So matching those together is super hard. And if you break them with this set of restriction enzymes, your gel is going to be pretty much the same everywhere, because at this point, the sequence is not big enough to produce highly separated fragments. So if you take your 100 KB, you cut it with 3 restriction enzymes, you can get a different banding pattern. But if all your fragments are 600 bases long and you cut it with those restriction enzymes, then you'll have a lot of 200 bases, a lot of 300, a lot of 400, and it's very hard to differentiate. Okay? So that's pretty much the answer to this. Next lecture, we're going to talk about the actual sequencing that was done for both projects, and then get into the different techniques if we have time. Okay? Anybody coming to the office except Gabrielle? Okay, cool. So I'll see you next week. I'll correct your stuff, finish correcting this weekend, and send it to you so that you can kind of see my correction style. Obviously, I'm not going too hard on you guys, so it should be good. And then don't forget to submit your assignment for next week, okay? Good, so take care.